Good morning, everyone. Welcome to the webinar for December. My name is Rebecca Bailey, and I will be the moderator for today's webinar. Before we start the webinar, we do have a couple of housekeeping items. The first is that a recording of the webinar and the slides from the webinar will be posted in the webinar archive site after the webinar, along with a link to the ATM model publication. Please give us a couple of hours after the webinar to process the video before looking for the recording. And second, the speaker will be taking questions at the end of the webinar. You can ask questions at any time during the webinar by entering them into the questions box on your GoToWebinar side panel. Today's speaker is Dr. Norm Mazur. Dr. Mazur is a hybrid, an MD physicist mathematician with over 35 years of experience in pharmaceutical R&D, medical affairs, and academia. As a disease modeler at the Roche Innovation Center in Basel, Switzerland, Dr. Mazur developed semi-mechanistic mathematical models related to diabetes, reverse cholesterol transport and atherosclerosis, macular degeneration, hemolytic diseases, and neurodegenerative diseases. His recent publication on the quantitative ATN model links amyloid to clinical outcome in the pathogenesis and treatment of Alzheimer's disease. Dr. Mazur's interest in mathematical modeling began during the 1970s as an MD student in Harvard MIT program in health sciences and technology, and a Ph.D. student in the physics department at MIT, where he developed quantitative models of micelle formation, biliary lipid secretion, and cyclic neutropenia. Prior to joining Roche, Dr. Mazur was an associate professor of medicine in endocrinology, diabetes, and nutrition at Boston University, and was previously employed at Theratech, Watson Laboratories in Salt Lake City, and at Sandoz in Basel. He was also an adjunct professor of pharmaceutics at the University of Utah and a visiting professor in the pharmacy departments of the University of Basel and Swiss Federal Institute of Technology in Zurich. Dr. Mazur is the founder and sole employee of NAM Consulting. He's an advisory member of the Critical Path Institute and a scientific advisory board member of the International Degenerative Diseases Research Center. And it is with great pleasure that I introduce our speaker for today, Dr. Norm Mazur.
Thank you, Rebecca. Hello, everyone. For almost a year, I've been looking forward to sharing our work on the Q-ATN model of Alzheimer's disease in the ROSA webinar series. By the end of this hour, I hope you will have a better understanding of what these hieroglyphics mean about the mechanisms of disease and the effects of anti-amyloid treatment. I hope you will also appreciate that the Q-ATN model is still a work in progress. Let me begin with acknowledgments to my former Roche colleagues, to other workers in the AD field, modelers, and especially to you, Rebecca and Christina Friedrich from ROSA. Well, what is Alzheimer's disease and why do we need a model? I'm paraphrasing some of the statements on the Alzheimer Association website. Alzheimer's disease was described in 1906 by Alois Alzheimer, a German psychiatrist and neuropathologist. It accounts for 60 to 80% of dementia cases, and dementia is a general term for loss of memory, language, and thinking abilities. The majority of patients with this disease are over 65. There's a younger onset associated with rare genetic abnormalities. Typically, the patients live 4 to 8 years after diagnosis. Some can live much longer. And the symptoms progress from what is called a mild cognitive impairment to marked interference with daily life and sadly death. Amyloid plaques are clumps of aggregated beta amyloid protein that form outside of the neurons. And this is thought to be the initial pathological species in AD. Tau tangles are twisted fibers of aggregated tau protein that accumulate within neurons. And they spread from the medial part of the brain to outer cortical regions of the brain. Lastly, neurodegeneration represents the loss of neurons, the loss of synapse, the death of neurons, and the thinning of the brain cortex. Now, these three elements lead to dementia. Now, how is dementia quantified in clinical studies? Well, I don't expect you to read this, this table, but we are referring here to the clinical dementia rating sum of boxes, which I will call the CDR-SB. And this consists of 6 different areas: memory, orientation, judgment and problem solving, community affairs, home and hobbies, and personal care. And each of these areas are graded on a 3-point scale. And when this is summed together, we can have scores that range from 0 to 18 points on the CDR sum of boxes. Sorry there, okay. Well, why do we need a model of Alzheimer's disease? This slide is one of the reasons. It shows the time course of the CDR sum of boxes over a 300-month period that was compiled by Kim, and these dots represent individual patients moving along this trajectory with time. A second reason, and really the motivating reason for our work, was to try to understand why removing amyloid plaque from the brain could result in improvements in clinical status. And this slide, this figure, was actually taken from the FDA's review of another anti-amyloid compound, aducanumab. And what it shows is that as in different trial limbs and studies with different molecules and doses, a correlation between the removal of amyloid plaque going from right to left and the improvement in clinical status using the CDR sum of boxes and the difference between the treatment and the placebo, which becomes a negative number as the treatment improves. Well, what accounts for this? This is what our model tried to help us understand. Before getting to that, let me just share a few personal thoughts on models and modeling. Well, what are models? And here I will show a few thoughts. The first is from a wonderful book by McIntosh and McIntosh, who are endocrine physiologists, and they write that a model may define, behave like, or explain the workings of a system. Mathematics offers rigorous methods for testing hypotheses by comparing models with experimental data. And you'll see that this is really at the center of our approach. Well, something I learned when I came to the pharmaceutical world was that all models are wrong, but some are useful. It used to rankle me, but it's grown with time, and I appreciate the sentiment. And lastly, one of my heroes, Einstein, is said to have stated that a theory or in this case, a model, should be as simple as possible, but not simpler. So now, what is modeling? This figure was taken from a paper that my colleagues and I wrote about 10 years ago in regard to lipoprotein metabolism. It was based on the McIntosh and McIntosh book. And what it shows is that modeling and experiments both are derived from ideas that we have about how a real system may behave. This then gives rise to both experiments and an in silico model. From the experiments, we generate observed data, and from the model, we generate predictions. And then these are compared with each other. When that's done, the results may lead us to generate more ideas, and that can lead to further cycles of this process, a second or a third or even more times. And it's in this paradigm, modeling and experimentation are linked together in a cyclic process that ultimately leads to the refinement of the model and the ideas and the assumptions about the system on which it has been built. So now let me give some further background about an anti-amyloid therapy, an antibody called gantenerumab, developed by Roche, and the two pivotal studies, the Graduate 1 and 2, that were carried out in phase 3. Well, this is going to show you a cartoon model of the presumed mechanism of action of gantenerumab. And this is the IgG molecule with an active Fc fragment. The molecule binds to this clump of spaghetti as well as oligomers and monomers. The spaghetti is, in fact, amyloid plaque. And by the attraction of the Fc fragments, microglial cells in the brain end up binding to the plaque through the antibody and ultimately ingest it. And that's how it is thought that this molecule and this class of molecules generally operates. Now, this is a schematic diagram of the graduate studies. I just want to make a few points that the drug was administered by subcutaneous injection, that it underwent a titration period over the first 32 weeks of the study, and then when it hit the target dose, that was maintained until week 116. The primary endpoint of the study was the CDR sum of boxes at week 116, which represents also 27 months of treatment. And during the study, amyloid and tau were also assessed in separate PET sub-studies. Now we're going to start with a high-level view of the Q-ATN model, and this is the one that is published in the reference that is seen here and that you'll be given later. We're going to call this version of the model version 1.0. And as the name suggests, the model was based on a literature concept called the amyloid tau neurodegeneration biomarker research framework, which was developed by Clifford Jack and colleagues. To illustrate what AT and N represent, we have these lovely images from the Rabinovici lab, which show the amyloid PET appearance in the blue here. They show that at the same time in this patient, the tau PET is not everywhere, but is only located in certain parts of the brain. And when it was studied longitudinally, they found that it was in this part of the brain, same as the tau PET, where the brain atrophy was greatest over time. So this is one snapshot that shows a near longitudinal projection. And It is this so-called amyloid cascade that goes from A to T to N to cognitive impairment that we will formulate in the Q-ATN model. So here we have what we consider the 4 linkages in the model. And this panel represents how anti-amyloid therapy impacts the amount of amyloid PET. And it's a little hard to see in this image. We'll go into it in later slides, but you can see that there's an input to the growth of plaque, which has a parabolic shape shown here. Then there is a PK/PD component that describes the effect of treatments. And in the PD sense, we are hypothesizing that there's a first order elimination process on the amyloid plaque, which is proportional at any moment in time to the plasma level of drug. And this, and together, this leads to the treatment effect shown here over 2 years for a particular anti-amyloid antibody. Now, the second linkage is a bit more complex. It is the linkage between amyloid PET and tau PET. And according to the model, we hypothesize that amyloid, the level of amyloid plaque, governs or determines the production rate of what we call excess tau, which is the aggregated or pathological tau PET level. And we represent this with a family of curves representing the individual variation in this process. And this input rate leads to a series of curves that are shown here, which reflect the relationship between the excess tau and the amyloid PET level. The lower level here is what we call basal tau in the model. In addition, we hypothesize that there is a first order elimination process of the tau PET, and that this is characterized by a rate constant k tau. Now, this is a simpler linkage in the model between the tau PET and the cortical thickness, particularly in the medial temporal part of the brain. And what we assume in the model is that the excess tau determines in a proportional way the rate of cortical thinning, so the derivative of the cortical thickness. And finally, the last linkage is between cortical thickness and the CDR sum of boxes. And we assume in the model a very simple idea that as the cortical thickness decreases due to the disease process, the score, the CDR sum of boxes, keeps increasing until it reaches the maximum plateau value. Now, how do we calibrate these different linkages in the model? The first case here shows the amyloid input function, and there were 3 similar studies carried out by different research groups in which this inverted parabola was the best representation for the relationship between the rate of amyloid increase and the baseline amyloid level, with, as you see, lots of variability. Well, we performed a type of meta-analysis of these studies, not using the individual data, which we didn't have access to, but in all cases, but simply taking the 0 points of the parabola, averaging them, and taking the maximum height of the parabola and averaging it. So this green curve drives the model that we'll be discussing. And if we integrate that parabola over time, we can see that it predicts that the amyloid level, which we call A beta as a variable in the model, increases sigmoidally in time. And when we use the model to simulate a study, we have to integrate the whole model, all of the equations, for a period of T start until we get to the amyloid level that the study begins at. This may hopefully become clearer later on. Now, the other part of linkage 1 to calibrate is the exact kinetics of plaque removal. And here we illustrate that for the gantenerumab molecule based on the studies that Greg Klein reported in the open label extension of the Scarlet Road and Margaret Road trials. And in these different subpopulations that receive different amyloid dosing regimens, we see that the Q-ATN model can describe very reasonably the mean behavior of the amyloid PET. And this is the value of the alpha REM parameter that we derived for gantenerumab based on these individual studies or individual cohorts, one value. Now, the calibration of linkage 2 involves the dynamics of tau PET, and it was based on longitudinal data of tau and amyloid from the Harvard Aging Brain Study that was reported by Keith Johnson and Risa Sperling in 2020. Here is their data representing the tau PET levels in the entorhinal cortex and the inferior temporal cortex plotted versus the amyloid PET levels in individual subjects. Now, these arrows represent longitudinal data in the individual, where the beginning and the end of the arrow represented a median of about 2.5 years of observation. Now, we looked at this data and characterized it by what we call the Basel tau level, shown as this horizontal line. And values that are above that, particularly in these regions, represent the excess or pathological tau. And these family of curves are derived in the model, and it is our way of trying to capture the variability in these arrows. And the different curves are shifted by a parameter that we call A beta 50, which simply moves them from left to right in the mathematical formalism that we've generated. Now, how did these curves come about? Well, there's a production rate which is assumed to be modulated by the A beta level. So however much amyloid plaque there determines the rate of production of the excess tau, but with variability according to the A beta 50 parameter. And we further assume that the tau PET aggregates can also be eliminated by an endogenous process that we characterize with a rate constant k tau. Now, there wasn't much data to estimate this when we developed the model initially, and we just took a value that was based on very limited preclinical data of 0.5. Now we look at the calibration of linkage 3, which is the relationship between the tau PET levels and the rate of change of the cortical thickness in the entorhinal cortex here, and the medial temporal cortex in the other slide here. And these green lines represent our more or less mean behavior reflecting this part of the model. You can see that at the tau 0 point, which is when we're still at the basal level, there is no rate of change of the cortex, but as the excess tau increases, the rate of change increases. And we estimated this slope from these and other data sources to put into the model as a fixed parameter. Now, the last part of the calibration involves linkage 4, which is the relationship between the medial temporal thickness and the CDR sum of boxes. And again, there was relatively little data to go on while we were developing this. There were 4 data points from different populations that Brad Dickerson's group had reported, and we used our mathematical relationship to fit these data. We also used longitudinal observations that studied the progression of the disease to much higher levels of CDR sum of boxes than we saw here. And by solving the whole model and estimating the parameters of this relationship, we were able to define reasonably well this blue curve, which is the relationship of linkage 4 in the model. It's basically a inverted hill function that's capped at 18, and we estimated that it starts deviating from this point, which is the cortical thickness without any deterioration, and the Emax and these parameters were derived from analyzing this data. So now, having calibrated the model, let's try to look at some validation efforts. And the first one concerns longitudinal studies of tau PET that were carried out by Jack's group. Here we show the simulation of how the rate of change of tau PET should depend on the amyloid beta level, that's the A beta parameter, during the natural history of the disease. And in this sense, it doesn't depend on our parameter k-tau. The natural history part of the model is invariant to that. And when we compare these simulations, which are done with different A beta 50 levels that sort of characterize the middle of the populations that we've looked at, we can compare that to Jack's data. And his data was in 3 small subgroups: a cognitively impaired amyloid positive group here in red. The blue is a cognitively unimpaired group with a lower amyloid PET level. And lastly, an unimpaired, cognitively unimpaired group with very low amyloid levels. And we see that there are no free parameters here, that we describe these data reasonably well. But we would like to have data at higher amyloid levels out here to see if we can actually illustrate and verify the declining trend shown in the simulation. Now, this is validation of the model using other studies, other natural history studies of the CDR sum of boxes. In the left panel, based on Delors' analysis of the ADNI data set, we see that the 2-year change in the CDR sum of boxes is itself a function of the baseline level. It increases and plateaus. The brown line is what they determine by the data analysis, and the green curve is predicted by the Q-ATN model directly with no adjustable parameters. On the right panel, you see the data that we showed earlier from Kim with a superposition of individual Q-ATN curves. And these curves simply start at different amyloid baseline levels, and they're all able to predict the kind of trends that are seen here across this population data plot. Now, a final validation of the model was based at the time on the aducanumab eMERGE study. And this shows in the upper panel how the amyloid plaque diminished with both a high dose and a lower dose treatment. And in the lower panel, it shows the CDR sum of boxes that were reported in the study. Now, here we show the Q-ATN simulations where we were able to predict, actually from phase 2 data, what the plaque removal would look like, and it matched very well with the observed data. And here is the simulation of the CDR sum of boxes, where we only need to tell the model what was the baseline level to simulate these curves, and it was in surprisingly good agreement. We did similar simulations for some phase 2 studies that were in the literature and with different treatment limbs, and we were able to show a fairly similar result to the FDA plot, which is that as amyloid is removed by the different treatments, the treatment effect on CDR sum of boxes becomes more negative, becomes more favorable. Now, with these validation steps, we went ahead to try to simulate what we thought would happen if we were to use gantenerumab in a 5-year study. Now, this is a hypothetical case. It is based on the treatment regimens which were used in the graduate studies, and after the 27-month endpoint, we simply maintained the target dose for 5 years. This panel shows how amyloid is predicted to decrease in the treatment group and increase slowly in the placebo group. Here is what the model predicts on tau PET. Here we show how the CDR sum of boxes will change, and it's delayed relative to the earlier biomarkers, but it eventually bends as the treatment slows the rate of progression. And finally, we look at what is predicted for the CDR sum of boxes in the placebo versus the treated group. We see a very large effect at 5 years, or we predict that. But the effect at 27 months is something that we were waiting to verify, if we could, with the graduate study results. So now, the moment of truth, comparing our predictions to the graduate 1 and 2 outcomes. Let's first look at the amyloid PET data from the graduate 1 study. These were our simulations over the duration of that trial. And this is the treatment data in the circular symbols. We predicted very well the placebo effect, or the natural history, but the drug effect was not as strong as we had estimated. And now we look at the CDR sum of boxes. These were our simulations, and now we show that the actual data. We predicted the placebo group very well. However, the treated group did not show much difference from that at all. And if we look at comparing what our simulation was to what the treatment effect was in its 95% confidence interval, we predicted a much bigger effect that did not fall into that interval. And naturally, we were disappointed. And naturally, the study itself was, in this case, a quite unfortunate outcome. Now, we had a second chance with graduate 2. These are the simulations of the amyloid effect removal. This is the amyloid results in the trial. Again, amyloid was not removed at the same extent and rate as we had predicted. Here we look at the CDR sum of boxes simulated, and here we show the observed data. Again, the treatment effect is very slight, and our prediction was much bigger and did not coincide with the data. Again, this was a disappointment. So, from disappointment, new thoughts, new ideas came, and we wanted to try to update the model to see if we could get a better prediction, or at least a better simulation. And as we looked at the data, we concluded 2 things: that the alpha REM parameter in the model was not correct, and that needed to be changed or re-estimated. And we also concluded that the k-tau parameter, the rate at which aggregated tau is endogenously eliminated, was not correctly estimated, and we wanted to use as much data as we could in a sensitivity analysis of multiple treatment arms to better estimate that. There were no changes to the other linkages. So here we look at the graduate 1 results. Again, we've seen this already, and now, we have re-estimated the alpha REM parameter, and we see that it's about 34% smaller. Here we have the CDR sum of boxes with the original k-tau value of 0.5, and this was what we saw before in terms of the treatment effect. And with the new k-tau, a smaller value of 0.2 from our sensitivity analysis, we have a result that is well within the confidence interval. And this was a much more satisfying outcome, again, after the fact. Here we did the similar thing with graduate 2. We re-estimated the alpha REM parameter. It was even smaller, or greater reduction in the graduate 2. And then here we have the CDR sum of boxes, and when we re-estimate that with the 0.2 k-tau value, we get a result that is again within the confidence intervals. So we are happier with this result. Now we have also used the updated model to look at another molecule, the lecanemab molecule, which was reported about the same time a year ago. Here was the amyloid PET data observed, and what we had previously simulated from the phase 2 study. We had to likewise adjust that alpha REM just by a bit to get better agreement with the data. And this is the CDR sum of boxes. It's a little bit off in terms of the slopes. The prediction was on the high side with the original k-tau value. With the lower k-tau value, the treatment effect is smaller, but within the 95% confidence interval. So that is where we stood with that study a year ago. Now let me just illustrate how we had determined this updated k-tau value. On the right side, you see that there are 7 different treatment arms from graduate 1 and 2, Clarity, and the 4 aducanumab dosing trials in phase 3. And our original k-tau value produced these outcomes with respect to the observations. Three of those values fell outside of the confidence intervals, which was not satisfying. And what we did was vary the k-tau, keeping track of the overall deviation from the observed treatment effects. And we found that a value of 0.2 minimized these deviations. So we used this data and got a new value for k-tau, and you can see that all of the treatment predictions fall within the confidence interval. And this is how we got that updated value. Now, as a further test of the updated model, which we called version 1.1, we looked at the reported data on tau PET from both the Clarity study and the aducanumab study. We went back and looked at that. This was the reported tau PET data from Clarity. It again was in good agreement with the placebo result, but the original model over-predicted what we saw. With the smaller value of k-tau, we seem to be in better agreement with that data. Here we show the tau PET data from the aducanumab study with the k-tau originally at 0.5 And then with the k-tau at 0.2, we had much better agreement with this small sub-study. Now let's look for completeness at what we learned with gantenerumab in its tau PET study. Well, this is what the model would have predicted. The updated model, which did a better job with the CDR, is still not in very good agreement with the tau PET reported for gantenerumab. And we think that there could have been a number of reasons that that study just wasn't adequate. There were many discontinuations over the study period. There was actually no significant difference in the CDR sum of boxes in the sub-study population, and no difference in their tau PET. And it could have been that the tracer that was used in this study, the GTP1 tracer, is different from how we've calibrated the model and may not have been adequate for what we needed here. So now let me move forward with this work in progress, because we've had some new studies that were reported since the updated model, and we're going to call this version 1.2. And there are two different studies that we're going to analyze now. The Donanemab phase 3 study called Trailblazer, and a different class of molecules, a tau-targeted antisense oligonucleotide, which is called BIIB080. That's a tongue twister. And there's data reported from the MAD study that we've taken a keen interest in. So first, let's talk about Donanemab and the Trailblazer study. This is just a schematic of the study design, and I want to emphasize just two aspects of this. The first is that there were actually two subpopulations recruited for the trial, and these were based on their tau PET baseline values, a low medium tau PET and a high level tau PET, and we'll look at their data separately. The second is that as the study progressed and as amyloid PET levels were being measured, if they fell within a range in which the amyloid had been cleared adequately, then the patients were put on placebo instead of maintaining the dosing that was previously given in the trial. So there were dose reductions, and that makes the simulation a little more complicated. So let's just first look at some baseline data in the two tau PET subpopulations, the low value and the high value. And I want to just emphasize a couple of features in this table, which was compiled from the published study of SIMS. First of all, the N values were about twice as large for the low-medium population than for the high tau population. And that was by design in the trial. This was not used as a primary endpoint. This population and the total population were, but we think it's useful to look at them separately in this way. Secondly, the ages of the two populations were significantly different. They were older in the low tau population than in the high tau population. The CDR sum of boxes were also different. They were lower in the low medium tau and higher in the high tau population. And although the amyloid PET levels were rather comparable, as we would have expected, the tau PET baseline values were quite different. And I want to mention and emphasize that this tau PET value is measured in the neocortical composite region defined in the Donanemab trial. It's not the same region as the medial temporal cortex that our model has been designed to represent. So these numbers are not in the same relationship to our simulation. First, let's look at how the doses changed. Dosing reduction could only begin after week 24 when the tau PET was measured, and we see that the dosing reduction was greater in the low medium tau group than it was in the high tau group. We'll try to explain why shortly. Now we're going to look at the estimates of alpha REM in the two subpopulations, again based on fitting the amyloid PET data that was reported. So this is the data that's reported. You see the small observations, and we have fit this by adjusting the alpha REM value to describe the data, and the value that we get is shown here, 0.0345. Now the high tau PET data is shown here, and you may appreciate that there's a slower reduction, and that's reflected by a smaller alpha REM in this population. I will explain these parameters F shortly, but the larger alpha REM seen here may reflect the older average age of these patients compared to these subjects. The F parameter is something that we use in the model to represent the fact that even though the mean doses are lower, the apparent removal rate process is slower in this part of the trial than it was earlier on. And this may represent just the more complex individual populations that are being represented by the mean. These values that we have estimated now for the phase 3 population are very similar to the ones that we had estimated from the phase 2 Donanemab study, and it's interesting to note that the age of the subjects is very similar there. And this idea that age affects the rate of amyloid removal has also been seen with aducanumab and lecanemab. Let's just now look at the CDR sum of boxes, and I want to mention here that what we're doing is estimating the k-tau value that gives us the best agreement with the treatment effect. So here in the low tau group, the placebo, which doesn't depend on the k-tau, is in very good agreement with the treatment, with the observations, and the treated group is in fair agreement. The difference is minus 0.68, which is the largest seen to date, and the k-tau, interestingly, is 0.58, even greater than what our updated value is. Here we look at the high tau group. The simulations don't show quite the very steep slope that was seen here. They're a little bit faster than before, but we've optimized the k-tau to match the treatment effect, which is similar in absolute magnitude as the other group, and the k-tau is nearly the same. So here we have kind of a new puzzle. Why are these k-tau values higher than we had previously estimated for Gant and lecanemab and even aducanumab? The last, here we want to look at the tau PET data itself. Now, first are the simulations for the Trailblazer study. This is the baseline value. This is what we expected the placebo group to do, the treatment group to do, and the treatment effect on tau PET. This is the other group, similar results in terms of placebo, treatment, and treatment effect. And now let's compare these results or these simulations to the observations. Well, here we have the low tau group, and we see that there is really just a slight worsening of the tau PET rather than the reduction that we expected. And here we look at the high tau group, and it's a very small reduction, which is much smaller than what we had predicted. Now, these discrepancies between the simulated and the observed could be due to a number of factors, and maybe the most important is the region in which the tau PET is being measured. In the simulations, it's the medial temporal cortex. In the observations, it's this neocortical composite. It could be that the floor tau sapere tracer is just not as sensitive to these treatment effects, which we have seen happen with the MK6240 tracer in the previous studies. And it could, of course, be deficiencies in the model. What we feel is needed is tau PET data corresponding from the study in this MTC region. I'm not sure that we will see that, but if it were reported, it would be helpful. Now, lastly, on the Trailblazer study, we just want to show what our simulations of the medial temporal thickness looked like. The placebo in black, the treatment effect in blue. And we see that if we express it in terms of the percent change, it's a measurable effect. Should have been measurable, but they didn't measure the thickness. What they measured was the hippocampus volume, and it shows something that's at least qualitatively similar. It takes a while to see a difference, and then it's a small difference. The treatment effect here is measured in terms of the difference in volume of hippocampus. We would need the baseline data, which has not been presented, in order to calculate percent changes, and they might have been of a similar magnitude as these. Now, in the last couple of slides, I want to show you the tau PET, the anti-tau oligonucleotide results. This is the MAD study, just been reported a month ago. I'm going to focus in analyzing one of the cohorts of this MAD study, which had the highest dose of anti-ASO treatment every 12 weeks administered intrathecally. And I want to point out that there's phospho-tau data in CSF, as well as tau PET data that was measured. This is the features of the study population. I just want to emphasize that the tau PET levels are much higher than we've seen in the other studies. So this is a group that has substantial tau pathology. It has a CDR similar to the other studies that have been reported out there. And this shows here the CSF tau, phospho-tau levels, which decrease very rapidly in the treatment group. These are the brown curves, and then they flatten after the MAD pretty well during the long-term extension. Interestingly, the tau PET data that was reported for cohort D shows very little change in the MAD period, but after that, it does show a decrease during the 75 weeks of long-term extension, and the decrease here is substantial, minus 0.6, much greater than any change that's been seen with anti-amyloid therapy. And one other feature of the data that was interesting is when one looks at the individual cohort subjects and plots their change from baseline as a function of the baseline level, there's a correlation seen. And if we use our model, we'll see how that goes shortly. I got ahead of myself. Now, using this data, we use the Q-ATN model or features of it to try to estimate k-tau. And we did that by looking at the excess tau over the tau zero value at these two time points. And by the simple formula here, we can estimate k-tau for each of these subjects, except for the one whose value passed below tau 0. So we have 7 individual values agreeing well with the data, and a k-tau value determined from this study, which is close, it's 0.489, close to the Donanemab value. And the model also can agree well with this data, showing that the treatment effect increases as a function of the baseline. This is explained by the first order elimination process. So I'm sorry that I've been speaking a little bit longer than I want to, but I want to end up with a couple of more slides. Is the Q-ATN model too simple? The answer is probably yes. More data are needed, particularly tau PET. And these are some of the improvements that one could consider. Differentiating in the L1 linkage between monomers, fibrils, and plaque. In the L2 linkage, we could look at the whole space-time evolution of tau PET, but we especially want to try to understand why the k-tau values are differ from each other as we've estimated them, and do they depend somehow on the rate of amyloid removal. The L3 linkage could be elaborated to look at other effects on neurodegeneration in addition to tau. And the L4 linkage could also be looked at in terms of different regions of the brain and how that maps to different components of the CDR sum of boxes, as well as whether the medial cortical thickness or the hippocampal volume would be useful in further developing this. And most importantly, a population model needs to be used on the available data to really get the individual parameter distributions and splice together many different data sets that could speak to this problem. Was the Q-ATN model useful? Well, that's up to the modelers in the field to decide. It is a semi-mechanistic framework that we've used to integrate and analyzing data in the field, and most importantly, it tries to explain how amyloid removal leads to clinical benefit. It's allowed us to simulate short-term and long-term effects on biomarkers. It's an evolving tool for supporting the design and development of future molecules and clinical trials. And it's a conceptual framework that could guide future research into the molecular and cellular processes in Alzheimer's disease. And it could be used as a reference model for future developments of empirical approaches like AI approaches to simulate this. What's my overall assessment? At the moment, the pieces of the model don't all fit together with the available data, and that's okay because the Q-ATN model is a work in progress. And now it's time for questions. I apologize. I'll stay longer. Feel free to ask them.
Thank you, Norm. We do have a few questions.
Okay, please go ahead.
Okay, so let me start. Regarding the parabolic amyloid function, is there any physiological explanation for the observed curve going up and then coming down?
We try to answer that in the supplementary material to our paper, so I suggest that you look there. There is an explanation for it. I don't want to go through that now, given that we have other questions, but check our supplementary material.
And in comparing your Q-ATN simulations with the Graduate 2 results, did you consider patient non-compliance as observed in the clinical trial as an issue?
Non-compliance is not a feature of any of these simulations, but since the models do simulate on the basis of the observed amyloid PET results, especially the revised and updated model, any type of missing doses would be accounted for in our estimates of alpha REM.
So can you use the model to explain the effects of the acetylcholinesterase inhibitors on amyloid plaque?
Short answer, not at this time. That's an area I have not looked at.
And after re-estimating parameters to allow the model to fit the graduate data, can it still fit the data from the earlier trials, your the road open label extension trials?
Well, there was clearly something different in terms of the alpha removal parameter in the open label extension and in the graduate studies. And so in that respect, the updated parameter would not produce as much amyloid clearance. But in terms of the other studies that we described in our first paper, I've shown how there are some improvements in the updated model in terms of the tau PET data from aducanumab, and we're still in the same range of the confidence interval for its CDR sum of boxes, though the updated effect would not be as spot on as the original one. So again, the model is very simple. The number of parameters and the structure of the model is fairly rigid and small, and we don't have any real flexibility as we would get in a population-based approach to really tweak these for the different studies. That's something that I hope will be done by interested people in the field.
Have you looked at other outcomes besides the CDR, or even looked at subsets of the input to the CDR?
Short answer, no. We have not looked at any of the other metrics that are used to capture cognitive function that some of the other molecules in the field have used. We focused on CDR, and we haven't really tried to break down the CDR into the different 6 categories. I mean, to me, it's amazing that an instrument like that, with qualitative statements that one assigns to numbers, is able to capture in a number the progression of the disease, and that this model can even describe how that single number grows over time with the intermediate processes represented in natural history and in the treatment effects. That's something that I think the model has helped us to at least think about and discuss.
I'm going to make a statement here for Ibrahim Mustafa. I'm going to send your questions to Norm directly to address them. I think they would be fabulous to follow up offline. I'm going to put in one final question from the audience, which was, how did you select that basal tau level or tau 0? What was the biological reason for that?
Well, it wasn't a biological reason. It was an empirical observation. We looked at the original data that Johnson and Sperling had presented, and they actually had averaged those 2 plots that we showed here in their original presentation, and our value of tau 0 was pretty compatible with the low end of where the tau data were. We further showed in writing up the paper that literature that had estimated the tau PET results with fluortalsapir in cognitively unimpaired populations was very close to this 1.15 value in tau 0. Now, with different tracers, with more data, that would be a parameter that could be different than the value that we had, and it certainly would have, at least hypothetically, a population variation, either seen in the unimpaired people, or when you have patients with manifest disease, we don't know what their tau 0 would have been, but it could have been different from a single value. So that's the answer to that question. I'm happy to take any questions that anyone has by email. I look forward to responding to you, and I thank you all for your patience and understanding in hearing a lot of stuff presented in this hour.
Thank you, Norm. Just a note to Carter, we'll forward your question on as well. I'd like to thank our audience, and I'd really like to give a big thank you to Norm. We've been waiting for this presentation. I know that much of it is unpublished, and you were thrilled to be able to actually get all of the data that you've been looking at. With great thanks, Norm, for finally being able to come on and present this work and talk to us about what you've been doing.
Well, I'm very appreciative, and especially of the Rosa Company for these seminars. And I can tell you that I'm glad that this will be, the talk will be given on the website soon. I will get the slides to you tomorrow so that that they can be posted as well. Because of the complexity of the builds, I have to do a little work to get them to you in a PDF file.
Great. Thank you, Norm, and thank you to our audience, and I hope to see everybody again at a future webinar. Thanks, everyone. Have a great day.
Bye-bye.
Bye.